to record. So in this example, if you counted the per residue performance, something such as this Q2 measure here that measures the fraction of residues correctly predicted in either of the two states, membrane or not, in this particular example here, you would see that immediately the majority of the residues are correctly predicted, right? You would have a very high Q2. So mispredictions are here, and here, here, and this little bit. But it's relatively few mispredictions. I also told you that a membrane protein is, the function of a membrane protein very often is revealed when you know the number of transmembrane helices that you have. You know that 7 transmembrane helices, a particular orientation, you find that protein, most likely it has to do has some receptor function, such as smell receptors. The bulk of those will indeed, in mammals, be smell receptors, right? On that note, how many smell receptors do we have in people? Do you have any idea? Just measuring the per residue performance is not the best way of doing it, because a variety of reasons. One reason is you're not actually capturing segments. And another aspect of Q2 would be exactly what you said before. When I say always non-transmembrane, on average in data sets I would do best, which is obviously nonsense. So we need a measure that punishes nonsense to be nonsense, and Q2 would not be that measure. So one measure would be to say that you correctly predict the number of helices. We have 3 helices observed here, and 3 helices predicted. In that sense, the always predicting non-transmembrane would be really punished here, because it would get 0 out of 3. But then again, this one is wrong too, right? You have the right number somehow for the wrong reason. How could you capture that? Well, what could you do? In fact, putting it somewhere where you have enough data and where making mistakes is not so bad is the best way of doing it. And then you can develop a method, and that method can sometimes predict that something is important to be rigid, and sometimes that it's important to be rigid, and sometimes that it's important to be flexible. So the same method can predict importance for 2 different things, and it's a classification that goes beyond conservation. And so it has, this was in fact a cover photo from Journal Proteins. It captures clearly the aspects of dynamics, and it captures some aspects of disorder, but it's not directly disorder. Okay, what is disorder? If I said that you shine light at that protein or that region, and at different time points it looks different. Going back to the NMR model, why are some, if you look at 3D structures, why are some regions more flexible than others? For functional reason. That is sort of one way of answering the why. But let's go back to the structural point. How come? What is it that makes them more flexible in the structure? Yeah?
Maybe they have more loops.
They have more loops. Ultimately, this is about contacts, so constraints. So the more a particular residue is constrained, meaning there are residues around it that it binds to, the more rigid it is. The less it's constrained, and the loop is exactly the kind of thing that is not constrained. That is why it's wobbly. That's because there are no residues around it that sort of constrain where it is, right? And so that gets you immediately to an idea how to predict disorder. You predict contacts or contact propensities, and then you simply look at regions that have very few contact forming propensity. So that look like they are not going to be constrained. So loop, in fact, would be one way, and I cut out this, our group, in particular, the first method that we predicted or that we developed simply predicted loops, very long loops, really secondary structure loops in the true meaning of that word. And when I say long loops, I mean things that are 100 residues long. And that you never find in globular proteins, right? That was our first way. Here is the first way that comes from Dostinani in Hungary, and they look at contact, but I don't actually have the quote here, which is a bad omission. I have only the method that Marco and Avner developed. Essentially, that predict contacts, and then look at low contact density to predict disorder, and that works too. Now, the important thing is, so far, I've spoken about two methods that capture something about disorder, or three methods. I've mentioned one, loops, that never uses disorder as a concept. One that uses B-values, very flexible residues, never uses disorder. One that predicts a lack of contact formation, only uses structures where you have regular structures that predicts contact. So none of those ever use the idea that there is disorder. Okay? And that is good, because if you develop machine learning devices and you develop them on data set without actually using the training set, or without using the data set for which you want to predict, that's the best way in which you can do machine learning. It's not always happening. And in fact, to move on here, unfortunately, we need to have some experimental handle on disorder, and that is complicated. And that is complicated because there is a great variety of disorder, and it's not easy to handle it. So the great variety of disorder, and that is Dyson and Peter Wright here, there is the kind of thing, there is a molten globule, so something that is sort of a little bit disordered, a little bit not. There is folding on target binding, meaning that you have something that while it's not bound, it's disordered. When it binds, it adopts a regular 3D structure. Then there are parts that are sort of mostly, in fact, disordered. And mostly disordered could have functional reasons. So you're not disordered because you essentially want to occupy space. This is indeed one of the reasons why things are disordered. So there's the idea, and the name is missing here. I do not know why. This comes, I call it the Dunker hypothesis. It's from Keith Dunker. And his idea simply was, when we look at three-dimensional structures, we see that there are some residues for which we have no resolution. No resolution means that essentially you don't have a signal in many crystals where the scattering of the X-ray beam is coordinated. That means something has moved, okay, between these two. And that means that something is very, very, very flexible. Essentially, so flexible that you don't get a signal. You don't get a measurement. And he argued that if I looked at the set of all of those, they would capture disorder. And that is a remarkable, he did that long before people assumed that disorder existed. And he showed that this sort of thing is very different. The signal is very different between different residues. And here's a photo of Keith Dunker, and developed a database with many others on it called Dispot. And it was a very visionary thing to do. Now this database now, unfortunately, has a limited size, simply because it's difficult to experimentally capture these proteins. And limited size, and you have a large interest in developing prediction methods, so everybody jumps on that and trains machine learning methods on that. And there is some complication coming out of that. Here's one of those, MD, meta disorder, that simply uses a bunch of different methods. Just using a bunch, you create a method that is already a little bit better by simply averaging, and you can then sort of do the averaging by sort of putting a bunch of methods as input, and some of these methods that are used as input predict B-values, some predict loops, some predict contact, a lack of contact, and all of this is put together to predict disorder, and it works, at some point it worked better than things that existed. But the problem really is it's trained on data, and the data set is too small. But anyway, so there are different aspects of disorder, and different methods find different aspects of disorder. I want to skip over that. The one thing that I want to sort of point out, if I asked how many proteins have some regions of disorder, and here from Dostoevsky the method IUPRET, and again the quote is missing, so this is a publication from Esmeralda Vicero and Avner Schlesinger, and it says that in eukaryotes, depending on whether you use IUPRET or MD overpredicts a little bit, IUPRET for this particular case may sort of underpredict, you see something that is sort of 36 to 43% of all the residues have some long region of disorder. Long means 30 residues and more. Okay? When you look at bacteria and archaea, this number is much lower. In fact, if you look at the difference between kingdoms, the number one thing in which they differ is exactly that. So much so that you can even distinguish between different bacteria. So the bacteria that live in heat have less disorder than bacteria that live in the cold. So essentially you can see that a sort of disorder or changing the content of disorder allows you to really change your environment or adapt to the environment. Keith Dunker is also behind the idea of more molecular recognition elements. So if now, and that is the idea, if eukaryotes have more disorder because they can do more complex biology with it, then more complex means more regulated binding. And then there must be regions, in fact, in proteins that are sort of disorder-like, and they are used to bind different proteins. So you have the same region that can adapt to different proteins. You don't need two proteins to bind to two proteins, you just need one region. So you can do more complex operation with that. So that's the idea, you sort of induce structure into something that doesn't have structure, and you sort of see that these more kind of things are much more in eukaryotes than in bacteria and archaea. So they're sort of complex binding. And with that, we leave it and go to the level of 2D. So we talked about 1D prediction, we talked about comparative modeling, 2D predictions. And 2D predictions come as contacts and distances. The first question that you may ask, is it important to focus on the strongest interactions? So strongest interactions would in fact be to predict contacting of beta strands. Beta strand pairing in fact is the strongest, on average, the strongest kind of contacting you have. Helix contacts are weaker. So you could possibly try there, and historically that's in fact what people did. I'm going to run over it, and I'm going to sort of go to the other point here. What contact prediction methods try to do is they try to predict long range as opposed to short range contacts. In a contact map, I cut out here the lower diagonal because it's symmetric. The contact map essentially in this particular case, red means that the pair of residues is within 8 angstrom of each other. Wherever you have a set of residues that is within 8 angstrom, you have a red point. Short range means essentially short in terms of sequence. So it's within I and I minus 10 and I plus 10. So within a window of 20 residues here, that is cut out and we call that short. And then there's long. So it's the difference between actually predicting some contacts within a helix or within this loop here, as opposed to predicting a contact between that helix and that helix. If you want to sort of fold a protein in a large scale, what you really need is this big, how the elements come together, right? So in particular, if you think that now secondary structure prediction methods reach over 80%, then ultimately all you need to do is take all these secondary structure segments and put them together. So have a few long-range contacts and arrange them. That's the idea. So long-range contact prediction, and the major tool that people have been using toward this end for many years has originally been called correlated mutations or compensated mutations. Today some call it evolutionary couplings. The idea is always the same. You look at a sequence, you look at an alignment. Here we have 1, 2, 3, 4, 5, 6, 7, 8 organisms aligned, and that goes back to something that Debbie Marks published first. And you see that at a particular position I, you have 3 organisms that have an R, then 3 others that have a K, and 2 that have a W. You see that the same 3 organisms have here a D, here an E, and here a V. And that sort of induces this idea that you have some correlated event. So the way I visualize this correlated event, I actually have a constraint, so I and J are coupled in the sense that they're bound, they're close to each other in 3D, and that anything that sort of, this bond here is the thing that is evolutionarily constrained to evolve. Meaning that if, say, in this particular case, we have RD, we have a salt bridge, so you break the salt bridge, at least you don't break it, but you change the distance between the residues. The K has a much longer side chain than the R, and you compensate for that by putting something shorter in here. So you still maintain the charge. Or you completely get rid of the charge and you have two very strongly attractive hydrophobic residues, valine and tryptophan. But you see that the same position has sort of the same event happen at the same organism, right? So there's a coordinated event, and the goal would be to sort of look at the alignment and get out all of these events and then say, whenever I see a correlation, I say those two residues are in contact. The very first paper that claimed that this is the case is here. Correlation, evolutionary correlation, increasing correlation, C beta distance. So what you want to have is that whatever has a high correlation is down here, and whatever has a low correlation is up here. So you want something like this, or something like this, or something. And it's unclear whether you see that or not. Okay? But what the paper defended is at least you don't have a random signal. It looks pretty spread, and this was, so there were within a year actually 4 publications on this, and these 4 publications had very different conclusions. This one here said there is a signal, and this one said that because when they looked at what would that imply in terms of contact prediction, they saw contact maps such as this one. So they saw examples where that's the observation, that's the prediction. Some of these predicted. You have very few that are predicted, but some of these are right. Okay? So this is not by chance. You could not reach that. It's maybe too few to be helpful, but the argument was there is some signal in there and we can squeeze it out. And ultimately, so the people behind that who sort of argue that way, historically in some sense won. Because now there's a publication, or there recently has been a publication from the lab of David Marks and Chris Under. The first author is Thomas Hopf, predicted in blue, experimental in gray, membrane proteins that were predicted here in 2012. For none of those an experimental data set was available. All of these are complete predictions, completely blind predictions. So they are not similar to any protein proprietary structure. For none of those comparative modeling could be applied. In fact, they tried, they picked explicitly in this paper, they publish 19 or 20 or some number of proteins like that. And those were all proteins for which no comparative model was available, no experimental data was available. And over the time, now this is 7 years ago, over the time for some of those experimental structures came in, and you see 6 of these here. I'm not entirely sure what the count is now, the count I believe is 11. And most of these predictions were essentially, so these are the first 6, and they look all remarkably good. The point is, you cannot, and here are a bunch of publications, first authored by Thomas Hopf, no, this is from Debbie Marks, that sort of brought in this idea. And the big breakthrough really is to not look at the correlated mutations the way they did before, but to sort of see the correlated mutations as a highest probability path through the entire protein. And then you essentially still have this noisy data that you saw that looks like random. But it turns out that if you compile the highest probability through as a path through the entire model, that sorts out some of these issues. Because suddenly, yes, there are others you can confuse it with, but if you have to choose one, and if it's the most probable one, then you pick the strongest, and that's the best predicted, and then the next best is actually something else. Anyway, so a bunch of papers here. for me, this is clearly the breakthrough over the last decade in terms of protein structure prediction. This is sort of an example where you can apply an old type of fashion neural network and you get something. So red is observed, black is predicted. For some cases, we get some signal that is right, sometimes over-predict and sometimes under-predict. But there is some examples where you can see that this is useful. For instance, you can predict folding rate, but as I meant, the end of the tunnel came from Debbie Marks and Chris Under using these correlated mutations. And this, in fact, is one of these contact maps predicted. And there are 2 proteins here. That is for the training set. I cannot remember, most likely the way it looks here, gray is observed and red is predicted. You have to remember that essentially on the diagonal they don't predict, so they sort of cut off ±5 around, so short range are cut out. So the real gray one they cannot predict because they don't want to. And other than that, most of the gray has a red corresponding to it, and essentially most of the red are right, so it's remarkably accurate, and that leads to these type of predictions that I showed you before. Again here, de novo predictions for important proteins, and here are just 3, and their disease involvement for which nothing else existed. So we can do that. Or some people, the group of Debbie Marks and Chris Under is a web server now. This is available. The point is, this is available for membrane proteins for just, 50 of all the membrane proteins out there. The point, I did not finish that point. I did not point that out. I showed you this slide. I said that the fraction of membrane proteins is similar between eukaryotes and prokaryotes. Roughly, every 4th protein is a membrane protein. Okay? So, in human, that means we have about 5,000. And I said that they can do this for 50 or 100 proteins, or 50 or 100 protein families. So we alone, in human, we have 5,000. So why 50 or 100 compared to 5,000 in human alone? Ultimately, the answer is we need, for this correlated mutation to work, the evolutionary couplings to work, you need huge alignments. Huge alignments means families with 10,000 and more members. And that still is a constraint. So for those methods, in fact, if you have 50,000, it's better than 10,000. It's not only that they need at least 10,000, or 10,000 is sort of something that they used. I can't remember what the threshold was for this publication here, but it's certainly much more than 1,000. They stopped modeling for 1,000. So you really need large numbers. While a family of the size 1,000 today is no longer an exception, 10,000 still is. And 50,000 this is why there are so few. It's a prediction method that works very well, but it's not available for every protein yet. Now let's go to 3D prediction right out of the box. One way of doing is through molecular dynamics. So you essentially look at the energy simulation. And in this one sort of remarkable thing is the platform called Folding at Home that was started by Vijay Pandey in Stanford, that currently has 100 petaflops and comes, according to Wikipedia now, as the world's fastest computing system. 100 petaflops is a lot. For those of you who do not know what that is big. And that actually is used to sort of, for instance, fold up proteins with molecular dynamics. Essentially, that's one thing. Another recent publication is from the Schulten lab, in which they show an atomic resolution hepatitis B virus structure is large. And the third one that I want to highlight, in some sense the most remarkable one, is this computer Anton. I already talked about it from the D.E. Shaw research, which is a special purpose MD machine, highly parallelized machine that is built exactly for that. For MD is an entire institute of people who built only Anton, I don't know what the level is now, Anton 3 or something like that. And here is a publication, a relatively recent publication, which they show that you can use the combination of low resolution 2D contact prediction methods with cryo-EM data and essentially Anton and get very, very good structures, or remarkably good structures out of it. Again, this is something that is beginning to affect the world of drug development. So on the level of pharmaceutical development or drug development, this comes to a point where 3D predictions are now available. Okay? For proteins, I said they are available for comparative modeling, but outside of comparative modeling we cannot do it. Now for some proteins we can do it. That's what this statement is. Here is another set, December 2018, just 7 months ago. Google's DeepMind, AlphaFold, is a method that won the CASP13 competition because it predicted structure better than any other group. And this, in many years of CASP, has been the first sort of newcomer approach. In fact, it is Google's DeepMind, but there are important people from the CASP community, including David Jones underneath. So there's a lot of expertise from the structure prediction community in there. But that some sort of newcomer ever won in the first run. This has not happened in many years. On the other hand, so for many of the predictions, there was a model from some other prediction method that was better than the Google one. On average, they got the best result. And we don't quite know how it was done. The talk is available from the engineers. We do know it's deep learning, and we do know that there's a lot of muscle underneath. So there are many things done that if you sort of understand deep learning, like some people in our group, then you realize as a university, if we worked on it for the next 10 years, we may have 1/10 of what these guys pumped in there in a week. So this is an example where, in fact, in a research, Google is completely using its muscle to make things possible that are very difficult to compete with. How far that now really is, method is not published, and the method is clearly not available. So will that in fact be applicable to every protein? And will there be a run for every human protein? Does Google have any interest in that? We don't know. So much of that is hype, how much of that is reality? At this point we don't quite know. But I believe what it clearly shows is that when I have given this lecture here in the years before, I typically tended to argue that we are still not there, and it seems that it will take a while. I believe we are at the point in 2019, we are for the first time at a point where we may hope that over the next few years, something new is going to come.